Hello, everyone. Thank you for coming to my poster. The title of my poster is Serial Measurements of Systemic Myokine Levels in Exercise Foals, a Pilot Study. My name is Samantha Hammack, and I am a DVM PhD student candidate over at the University of Illinois, Urbana-Champaign. Now, the clinical motivation behind this study is that racehorse deaths remain a significant welfare concern. They're still occurring around the world. Typically, these deaths are caused by catastrophic breakdown injuries resulting from musculoskeletal fatigue or microcracks. You can see a microcrack here in this image in the subchondral bone. And unfortunately, these microcracks are not typically caught on standard imaging such as x-rays. And also, racehorses aren't generally x-rayed before competition, so they're missed. The horses go on to exercise and they accumulate over time and the bone fails. Now, we know that bones remodel in response to exercise. In this study, you can see that a pitcher's throwing arm has thicker cortical bone than their non-throwing arm. Now, bones and muscles work together, and muscles act like levers to move bones. When the tendon pulls on the bone at the insertion point, it causes the bone to experience strain. At that point, the cells of the bones tell the bone to remodel, and that's when you get changes like this, where the cortical bone is thicker. That's the biomechanical aspect of bone remodeling. Now, there's also a biochemical aspect to bone remodeling that has recently been discovered. So it's been discovered that muscles produce signaling proteins known as myokines, aka cytokines produced by muscles, that can also interact with bone and encourage bone remodeling. The hope is that systemic or blood myokine levels may be useful as biomarkers of skeletal remodeling. In this study, I focused on three myokines, rank L, MMP2, and irisin, since they already are known to play a role in canonical bone remodeling pathways. There's previous literature on these three as well, and it shows that they do change with age and exercise, though the literature is limited. The objective of this study is to investigate the effects of both age and exercise on systemic myokine levels in growing foals using serial measurements. This is actually the first longitudinal study of serum myokine levels in juveniles of any species. So the overall study consisted of 12 foals, 6 control foals, and 6 exercise foals. The exercise foals were exercised at the age of 8 weeks for an 8-week period. The first week, they were exercised for 1,200 meters at around 3.5 meters a second. That's about a trot speed for people who know horses, or for other people, it's kind of like a fast jog for a person. In weeks 2 through 8, they went about 1,600 meters at the same speed, around 3.5 meters per second. This exercise study lasted for 8 weeks, so around 16 weeks was the end of the exercise study. Blood was drawn every 2 weeks during the exercise intervention, and then at the 20-week, 8-month, and 11-month time point as well. In the control foals, blood was drawn at times that were age matched to the exercise foals. Then, after the serum was frozen, and then in batches, sandwich ELISAs were performed that are specific to the three myokines mentioned, rank L, MMP2, and irisin. I then performed statistical analysis on the data. So what are my key findings? So the first key finding, is that there's no significant difference between exercise and control for any myokine. So if you look at this graph, on the x-axis, we have the age in weeks and then 2 months here at the end. And then on the y-axis, we have concentration, in this case in picograms per mil, because this is a representative graph. This is the graph of MMP2. And all three graphs for the three myokines look similar to this graph in that there were no significant differences between control and exercise group. Now, that could have been for any number of reasons. It could have been that the exercise was not intense enough for long enough. It could have been that the sampling scheme didn't catch any transient increases in myokine levels. So if the myokine increases in the hours following exercise, we didn't catch it because we drew blood at the same time every morning before exercise. And it may be that exercise may affect tissue levels of these myokines rather than circulating levels. Maybe we're having a more of a paracrine effect than an endocrine effect of these myokines. Now we're going to look So because the data didn't show any significant differences between control and exercise, I went ahead and grouped the data. And then I looked at changes over time. So as the foals aged, first I looked at rank L. Now, rank L did not change with age. If you look on this graph, again, age is on the x-axis, concentration is on the y-axis, and in this case, it's picograms per mil because that is what the kit measures it in. And you look at these 3 time points, all of which occurred during the exercise period, there was no significant difference. Now, this didn't we expected there to be a difference, so this was a surprise, but it's very possible that I just measured the wrong time points. For example, we might not have captured the plexiform to haversian bone transition at these sample time points. Foals have plexiform bone because they have bones that grow rapidly that is later converted into haversian bone, which is stronger. And we're not sure exactly when that occurs as the foals age. So it may be that we didn't catch that time point. I would expect rank L to go up around that time point because there's a lot of bone remodeling and rank L is associated with bone resorption. Next, we are looking at the systemic levels of MMP2 as the foals aged. And in this case, we did see an increase with age. So I had a significant Friedman's test for this data. And then I had about 0.07 p-value for this individual comparison. So between 10 weeks and 16 weeks, it does appear that the systemic levels of MMP2 did increase. This is actually as we expected. In the literature in children, actually not children, in elderly, that happened as well. And it's likely it's increased because MMP2 is involved in normal bone growth, and this was a period of rapid growth. Next, we're looking at irisin. So systemic levels of irisin, again, age on the x-axis, concentration on the y-axis, this time in nanograms per mil. I have the exercise period here highlighted in this orange color. For irisin, I saw several significant differences between different time points along as they aged. And overall, there was a decrease of irisin with age. Now, this is the same as what is found in the literature. In human children, they also saw a decrease with age. The reason that the likelihood is, though, that while irisin is mainly produced by skeletal muscle, it is also produced by adipocytes, and thus circulating levels may not solely reflect muscle activity. We may be looking at more metabolism and fat. Now, the limitations of this study, obviously there was biological variability between individuals, and we are limited to a smaller sample size because it's inherent to a horse study. And for future studies, work is ongoing to quantify myokine gene expression in muscle tissue, so I'll be able to localize and see if there is more of a tissue effect rather than a systemic effect. And in addition, additional studies with more subjects and intense exercise are planned. Thank you so much for coming.